Hello and welcome to Sci Made Easy. We are your hosts bringing to you complex scientific topics in a simplified way. Today's topic is cell division. There are two types of cells in nature: prokaryotes and eukaryotes. Prokaryotes have a single circular DNA, while eukaryotes have a compact condensed DNA inside the nucleus. There are three different types of cell division: binary fission, which occurs in single celled organisms like bacteria, yeast, etc., and multicellular organisms divide with mitosis and meiosis. In binary fission, the cell's DNA is first replicated. The cell undergoes elongation, following which it gets separated by a septum and it forms two different daughter cells. This binary fission is used for both reproduction as well as for growth. Moving on to multicellular organisms. Mitosis. Why do multicellular organisms need mitosis? It's because we require cell division for single fertilized egg to develop into a fully formed organism, for healing a cut, or even fighting off infections. Cell division happens all around us. It's very critical for life. Mitosis happens in the somatic cells of multicellular organisms, which are not responsible for reproduction. This phase is divided into 6 main phases. The first one is the interphase, in which the cell spends its most of its time. The cell grows, performs its normal functions, and prepares for division by replicating its DNA. The cell then moves to prophase when it has been destined for cell division. In this phase, the chromatin condenses into visible chromosomes and the nuclear envelope begins to degenerate. There are two polar structures called centrioles that also emerge during this phase. Then the cell moves on to the second phase of cell division called metaphase. During this phase, the chromosomes line up along the center of the cell, and this is called the metaphasic plate, which ensures that each daughter cell will get half of the chromosome number. Then the cells move to anaphase. In this phase, the chromatids are separated into two by the spindle fibers which are emerging from the centriole. This ensures that each cell has half the number of chromosomes that were present. Moving on, the chromatins start to decondense back into chromatin, and there are two nuclear envelopes that form again, creating two distinct nuclei. This is the final step where there is a split in the cytoplasm, and finally the one cell that we began with divides into two different daughter cells. Meiosis happens in reproductive cells. Meiosis is responsible for reducing the chromosome numbers into half so that after fertilization, the resulting cells has the normal number of chromosomes. The first phase is prophase 1. During this phase, crossing over takes place, which is the genetic recombination of genes within homologous chromosomes. These are chromosomes that are similar in shape and size. During this process, these chromosomes, they come together, and at this point a crossover occurs, and these are the resulting chromosomes that are formed. The cell then moves on to metaphase 1, where these homologous chromosomes align at the equatorial plate. Spindle fibers attach to the centromeres of these chromosomes, and during anaphase 1, these spindle fibers separate these homologous chromosomes towards the poles. Finally, the cells undergo separation and cytokinesis in telophase 1, where the nuclear envelope forms again and each cell has half the number of chromosomes. These cells further undergo meiosis 2, which is similar to mitosis, where the resulting daughter cells has half the number of chromosomes. Each cell undergoes prophase 2, then metaphase 2, where the chromosomes align at the metaphasic plate, followed by anaphase 2, where these chromosomes are connected to spindle fibers, and spindle fibers separate chromosomes into two chromatids. These chromatids then form two different cells, which are haploids, meaning they have 23 chromatids, and they are essential for reproduction. Now, for some food for thought, there are a couple of questions that we could discuss. How many daughter cells are produced at the end of mitosis? That's correct. 2. Mitosis leads to the formation of two different daughter cells. However, in meiosis, since we need half the number of chromosomes, it leads to the formation of 4 daughter cells. Moving on to the next question. In meiosis, during which phase does crossing over occurs? That's correct. Prophase 1. Prophase 1 is the first phase of meiosis, where two homologous chromosomes form a tetrad and undergo crossing over. Next question. Which structures pull chromosomes apart during anaphase? That's correct. Spindle fibers. The spindle fibers are fibers originating from the centriole that attaches to the centromere of chromosomes and separates them into two different chromatids. And the last question. Which of the following is not a difference between mitosis and meiosis? Number of divisions. That's incorrect, because in meiosis there are 2 divisions leading to 4 haploid cells, while in mitosis there's just 1 division that leads to the formation of 2 daughter cells. Genetic variation. That's incorrect as well, because in meiosis there are crossing over that ensures genetic variation, while in mitosis there is only division of cells. Involvement of spindle fibers. That's correct. Both division processes require the role of spindle fibers. Number of daughter cells produced. As you might have guessed, that's incorrect, because in meiosis the resulting cells are 4 in number, while in mitosis they are only 2. Thank you for listening, and stay tuned for our next video.